We're going to kick off this next round of talks talking about the safety that we are seeing in our patients that are on treatment. Lori?
Happy to be back up here. Obviously started this morning with talking about clinical benefit, but in clinical development, we don't just look at benefit. The first thing we actually look at is safety. We need to make sure that the drugs that we use to treat patients are safe. And so I will focus on safety, and you'll probably be sick of the word by the end of this talk, but we want to make sure you really understand how we track safety in our clinical trials. So I'll go first over the uniqueness of our nano-rare patients as it comes to looking at safety, not looking at efficacy. I'll explain to you, hopefully I'll explain to you what clinical safety actually is, how do we measure this in a clinical trial, and then give you an overview of the aggregated safety data in the n-Lorem portfolio, I guess. I did want to start out with, Stan was worried that you wouldn't listen to the rest of my talk if I didn't start out with this slide. We have not had an ASO-related serious adverse event across the pipeline, so we can put that to rest. And then now, you can listen to what I want to talk about. So it is really a challenge to treat nano-rare patients, also not just from the clinical benefit side where we have to try and understand what benefit means, but also from the clinical safety side. And it's the same reason than what I presented before. People come with a unique combination of clinical and disease characteristics. So again, the severe disease phenotype, the phenotype may progress even when they're on treatment. Is this due to the drug? Is this due to the disease itself? They have advanced disease. They're on multiple meds. There is no interaction with normal meds and ASOs, so that's not necessarily a problem, but the other meds could have a side effect, and then we need to understand what's causing it. Patients are also going to experience disease-related adverse events while they're on trial. I mean, we cannot expect that we're giving the first dose of ASO and then nothing happens anymore to these patients. So they have ongoing disease, and we're trying to treat that. And then again, the lack of natural history data makes it that we don't always know what the progression of the disease would be anyways without the ASO. So we need to learn and understand this as we go. For pediatric patients, we kind of have like an extra challenge on our hands. These little people grow into bigger people, and so everybody has growing pains, and this will happen also in our nano-rare patients. Our patients will mature. They will have advancing intellectual capabilities. They will go through puberty. We all know that can wreak havoc at any time. There'll be emotional growth, social pressures. And so we all need to take all of that into consideration and really look at the data as we go along. And then again, all our patients are at, various stages of their disease. And so, it can be tricky to understand where they are and what's going on. But safety is really the cornerstone of our mission. We obviously want to help patients, but the first thing we track and we look at is safety. Because if we don't develop a safe ASO, we just add burden to a patient who already has enough issues. And so that goes counter everything we would think of. With that comes responsibility and accountability on our part. So that's why we have to constantly monitor for safety. If there's an adverse event that we don't understand, we want to do mechanistic assessment and really try to understand what's going on. And I'll actually have a case for you. We'll talk about this in a little bit. We report the safety on a quarterly basis to our Data Safety Monitoring Board. So our Data Safety Monitoring Board is a group of external advisors, and I'll go over that too. We have to give updates to the FDA on an annual basis, and if something happens during a trial, we have to report to the FDA as well. We want to make sure, obviously, that we inform our investigators about anything that happens in one patient, in another patient, with one ASO, with the same ASO. And then we want to share information with the wider community. And so this is also part of that, part of the puzzle. So like I said, we monitor safety, and it's very important to us. We have a large group of people at n-Lorem who know how to monitor safety in a clinical trial and in patients with rare diseases. We also have the Data Safety Monitoring Board that is chaired by Eugene Schneider from Ionis Pharmaceuticals, and that is made up of external people, so non-n-Lorem people, external people who will review all the safety data and help us make the right assessment on whether the benefit-risk ratio is there. We are lucky, though, that we can really base our work on some solid foundation that has been built. in terms of ASO understanding, right? As Stan mentioned, Ionis was created in 1989 by Stan, who started n-Lorem. So we already have the wealth of information that comes right there, right? Ionis has treated thousands of patients in clinical trials with ASOs. But the most important thing that Ionis has done is that they have published databases of integrated safety data. And they've done that for the non-human primate data all the way to all the clinical trials. And so we know what to look for. And we also understand ASOs given systemically, intrathecally, and so forth. And then, of course, because Stan is our boss, we do continue to advance antisense technology at n-Lorem, and we really do try to take everything into account and make sure that we understand, everything that's going on in other ASO trials out there to really be able to take that information and understand what's going on with our trials. So an ASO is just like any other drug. There's desired effects that happen and undesired effects that happen, and that's all dose dependent. So we want to be in that window in between that we call the therapeutic index, where therapeutic effects happen and where toxic effects don't happen, right? But this is just the nature of any drug. If you push the dose, you will get toxic effects. So we really try to maximize the separation between the dose at which you receive benefit and the dose at which you receive side effects. That's why we want to make sure that we have, for allele selective ASOs, we want to have a really high selectivity, fold selectivity between knocking down the mutant allele versus the wild type allele. And so that's very embedded in all that we do every day. And based on past experience with ASOs with the same chemistry, we know what the right dose is, we know what the right route of administration is, and we know how to dose, like the dosing frequency. And like I mentioned earlier, we dose escalate slowly in every patient we treat. It doesn't matter if this ASO has been given to another patient already, we will still start at the low dose and slowly dose escalate. And we also know, based on past experiences, again, that a lot of the work that Ionis has done, what potential adverse events to look for. Right? So that historical and clinical safety data helps us look for the right adverse events. So for medications that are given intravitreally in the eye, we look for inflammation or cataracts. For medications that are given subcutaneously, we look for flu-like symptoms, injection site reactions, liver function tests. And then for drugs that are given intrathecally, we look for brain inflammation, hydrocephalus. And we know how to assess those adverse events. Again, because it's been done before. So we'll do regular physical, neurological, and retinal exams. We have established laboratory values for blood and CSF samples and various analytes that we analyze. There's MRIs, there's eye imaging that's done when needed. And then for some diseases, we're lucky, like for ALS, we're lucky that we can track neurofilament light chain. And so if we see a reduction, we see that something's good. If we would see an increase, we'd know something is going on that we do not want to see. But I do want to state that neurofilament cannot be used in every disease because the lack of natural history data, again, we don't always know what the levels of neurofilament are in a disease. And so more research needs to be done on that. So for every patient and every patient family who's here, I just want to kind of walk you through what happens and how we look at safety across the trial. And I really want you to realize it takes a village. We appreciate, like Sarah said, all the work that you do and sacrifices you do to come to the sites, but they're really important for us to be able to track the safety data. So when I work with a physician on the protocol, we make sure that we have the right safety measures in there. We have rules in the protocol on how to monitor safety. Obviously, the FDA and the institutional review boards will review the protocols, and so, you can feel good that it's not just people from n-Lorem who say, like, yeah, this is fine, but it got the approval from the FDA and from the IRB. And then, before we even start treatment, the n-Lorem clinical team will train the clinical site team on the protocol, the timing of the assessments, how to prepare the drug properly, how to collect blood and CSF samples properly. Then at the baseline visit, some imaging will be done, safety blood tests will need to be done, physical and neuro exams need to be done. That is really all because we need to have that baseline data. We need to know what the gestalt of the patient is before we even start the treatment. And then the day before the first dose, that could be around the same time as the baseline visit, but if not, then we'll do some more blood tests just to make sure, neuro exam, physical exam. On the day of the first dose, you may not realize, but everybody at n-Lorem is on standby. We know when the first dose happens for every patient. We care about the second dose too, okay? But the first dose is really kind of like the one where we just wait. I always try to tell the site, like, please call or like email at the end of the day, say everything went well. But we do give them space because they have a lot of work to do. But if we don't hear anything by 5 p.m., we do actually send an email like, all good, you know. But again, we just want to be on standby if anything happens. And sometimes it can be something very minor, like we know the pharmacy will call us because for some reason they want to make sure that we can release the drug. And you know, if it's at 7 a.m. Eastern time, I just make sure that Julie gets the phone call and not me because that's like in the middle of the night, but you know. Post-dose, if it's an intrathecal injection, patients have to stay overnight. For subcutaneous injections or intravitreal injections, you know, patients stay at the site for a little bit, and then when the physician deems it okay, they are released to go home. And then we will have regular calls with the physician to check on the patient, to talk about, you know, dose escalation, to talk about, you know, what they're seeing, what they're hearing from the families. And so we record all of that because we just want to make sure that we really track safety through and through. So what is safety? I keep on using the word and I don't define it. So in a clinical trial, safety is defined as the systematic evaluation, monitoring and documentation of any adverse event. That's the key term. Risk or unintended consequences that's associated with a drug when administered to humans. It's a mouthful. Every adverse event is described based on 2 qualifiers, and I'll go over the details of that. The severity of the adverse event and the relationship to the study drug, so the relationship to the ASO. What does that mean, really? First, I want to say that adverse events can be complicated by the intercurrent events that happen in a patient's life, right? So the adverse event itself is the unfavorable symptom, sign, or disease that occurs during the time that the patient is on study drug, and this can be just natural things that happen in your life, right? Again, intercurrent events. So we start collecting adverse events from the time of the first dose to the end of the study. Now, that is the end of the 2 years of the protocol, but that's really actually a lot longer than that, because as long as the patient under research IND is getting treated, we have to report the safety to the FDA on a yearly basis. So we will keep track of the safety for a long time. So the intercurrent events that can happen, and we have to collect all of those, whether we think they could be related to the drug or not. Could be worsening of disease symptoms, that could be the disease progression itself. It could be any infection. You know, patient gets COVID, we mark that down. You know, a patient has an adverse event from another drug, we mark that down. A patient ran into the table, fell on the floor, bruised their head, we write that down. So everything is collected in a clinical trial. How is severity defined? So we have, first of all, severity is how much the adverse event interferes with your daily activity. So you could have a bruise that is massive on your leg. That's still a mild adverse event because it's a bruise. It's not impacting your daily life, you know, so much. And it's really also the physician that determines the severity of an adverse event. So it's not anybody at n-Lorem sitting far away, right? It's the physician talking to the patient, and they determine what the severity is. So something can be mild. That is, it's easily tolerated, doesn't interfere with your daily life too much. Allergies, big bruise. Just looks ugly, but you know, not a big deal. Moderate. Okay, you broke your arm. That kind of results in some discomfort, but you know, you can still do most of the things that you do. So, but it is still not fun. Severe would be something that incapacitates you and interrupts your daily life. Imagine norovirus infection. You just need to be home close to the bathroom because you just can't go anywhere. How is relationship to the ASO defined? Again, we have 3 criteria for relationship. And again, this is the clinical decision of the physician, right? So this is not anything that n-Lorem decides when it's reported. So an adverse event could be not related. So there is no temporal relationship to the administration of the ASO. The patient got their dose a month and a half ago, they have a cough, the next dose is not for another month and a half. Well, likely they got some virus, right? So not related. It could be possibly related. Now, possibly related, if there's a reasonable temporal relationship, could be explained by another reason, but you can't really rule out the ASO, you don't know. So a patient got their intrathecal injection, they might feel nauseated when they wake up from the anesthesia, could be the anesthesia, could be the ASO. To be on the conservative side, the physician will say possibly related to that, right? And then we have events that we know are, definitely related. Let's say for subcutaneously injected ASOs, if you do get local red swelling at the injection site, then there's a strong causal relationship to the ASO being given, and that's the ASO would cause that. But the adverse event could also be related to the administration procedure, so the injection itself. And so it wouldn't have anything to do with the drug, right? And so you can imagine, again, for subcutaneous administrations, if they kind of nick something when they do the injection and you get a bruise or you have pain from the needle, that's pain from the needle, not pain from the ASO necessarily. Same thing with intrathecal injections. You can have lumbar pain or headaches that are LP, they're called LP headaches. It's the headache from the procedure, but not from the ASO. And then for intravitreal injection, it's the same. Pain or inflammation can actually come from the procedure itself, not from any drug that is in the syringe. And relationship is assessed just like the relationship, to the injection is assessed just the same as to the study drug. So not related, possibly related, definitely related. So when a physician has to assess an adverse event, they first have to say, is it mild, moderate, severe? And then they have to say, is it related to the study drug? And is it possibly related to the injection, all right? Just want to make sure everybody's on the same page with that, because it's important. So we also do have mitigation strategies that we can use to prevent discomfort. We can, use smaller needles, pre-treat patients with some medications, or we have post-dose guidances also on how to make sure that you have less LP headaches, for example. So now, what is a serious adverse event? So serious adverse event is different from severe. A serious adverse event is actually a regulatory term, and it's an adverse event that, in the view of the physician, meets any of those criteria. Results in death, is life-threatening, requires inpatient hospitalization or prolongation of existing hospitalization, or results in persistent or significant disability. So this is a different type of level of an adverse event, right? However, if you're in trial and you have to go into the hospital for an appendicitis, that will be collected as a serious adverse event. Not related, obviously, to the study drug, any study drug you'd be on, but it's related, we just have to report it, again. It's about the reporting and being completely transparent about the data we collect. If a serious adverse event is deemed possibly related or definitely related to the study drug, then n-Lorem has to file a MedWatch report with the FDA. And we have to do that in a very specific amount of time from the moment we hear about the SAE. So just wanted to be very clear about all of that. So safety oversight in clinical development has many different layers. We obviously have all our committees that help us make sure that we get to safe ASO from the beginning. But then we also have regulatory requirements to the FDA. Annual updates, we have to do that for every IND, and we have to send in safety reports if anything happens in a trial that deems that to happen. And you always have to look at safety assessment in the context of the disease, just like Stan and Sarah were talking before. So it's a benefit-risk balance of treating with this ASO versus not treating a patient. And oftentimes we know what the not treating leads to, and so we have to make that balance, risk-benefit balance. And that's really a discussion that we make with the physicians at every step. And so if something happens in a trial, the n-Lorem team will talk to the physician and we'll really discuss this to try and understand it best. So again, just a high level, when we have to inform the community of anything that happens, we have different types of communications for different audiences. The first people that have to be told is obviously the FDA. Then we want to make sure we let all the physicians know, not obviously the treating physician knows, but all the other physicians. We have to let the patients know. Sometimes we have to do an amendment to the informed consent if there is a new risk that has been found, and then we'll have to do that. And then we also want to, have safety updates, and we post those on our website. So recap of fundamental concepts here before I go into the data. It's very important, to have really good experience in ASO discovery and development and clinical trial to be able to track safety correctly. We treat every patient. So we treat patients that are maybe more mild, but we treat also the very severely patient and progressed patients. So we know that things will happen. And so we just need to be very transparent about it. We are tracking safety very diligently in every trial. This is a really close collaboration between our physicians and between n-Lorem. And again, adverse events that are unfavorable will likely occur during the study, but some of them are not related to the ASO, and some may be related to the ASO. And oftentimes, they can be related to the underlying disease of the patient. We have reporting responsibilities to the FDA and responsibilities to share the information with all of you. And so here we go. So our database has really started to increase a lot. Since I showed you earlier this morning, we're starting to treat more patients, and so we're able to aggregate all of that data. So as of this data cut, there was the data cut for the previous DSMB. We had 32 patients on treatment that are being treated with 18 different ASOs. Our first patient has been treated now for more than 3 years. We have at least 10 patients who've been treated for a year or more. And the age range of the patients we're treating, is anywhere from 3 years to 70 years. So it's not like a typical clinical trial where you would have a very homogeneous patient population. We have all kinds of people here. And we have a steady increased number of patients that we're treating because we have an increased number of new ASOs that are coming out of the lab and a lot of new patients that can use the same ASO that's already been developed, and so they can get to treatment faster. Again, a whole slew of different types of diseases that we're treating, so I think that's always very important to also keep in mind when we look at the data. And again, there's no ASO-related serious adverse event, but now let's get to adverse events in general. So the majority of the adverse events, and this is aggregated data across all our cases, right? So this includes most of it intrathecal injections, but also subcutaneous and intravitreal injection. So 58% of the adverse events we collect are not related to either the ASO or the injection procedure. And so that, again, that's the clinical, the investigator who makes that assessment, right? And so these are for ear infection, constipation, pneumonia, respiratory, any type of infection that they had or anything that happened. We have 15% of the events that are related to the injection or deemed related to the injection procedure by the investigator. So those would be lower back pain, soreness in back, that's normally from the LP procedure. Then we have 22% of adverse events that are deemed in that possibly related to either the study drug or the injection, right? And so remember, each adverse event can be rated possibly related to those two things if you don't really know which one it is, right? So take, for example, somebody who vomits after waking up from the anesthesia after receiving the ASO. You don't know from what it is. But if this patient, for example, always wakes up vomiting after any anesthesia, it's likely not related to the ASO. Or if it's the first time they get anesthetized and the first time they get an ASO, well, we don't know. But then the next dose they get, they don't vomit. And you're like, okay, so, we're good. But so that's why we are tracking these diligently over time, and we're trying to look if there is a safety signal we need to be aware of. And so that's what we did for one. We only have one, what we would call, it wasn't even deemed ASO related by the investigator, but upon further analysis by the internal team and talking with the investigator, we were like, well, maybe this is an ASO related adverse event. So what about that event? So this is an event that happened in our first KIF1A patient. So she's been treated for 3 years now, and she started to experience fevers post-dose for the last 6 doses. So she's received 14 doses to date. For the last 6 doses, she had fevers. I do want to state, because we're making this assessment, benefit risk based on treatment versus not treating this patient, right? This patient has shown significant clinical benefit since the beginning of treatment, and is still benefiting from this ASO. We also then look into the disease. What do we know about the disease? Okay, KIF1A patients, or KIF1A disease is associated with a lot of sporadic fevers in 50% of the patients. Well, we know these are not sporadic fevers because they happen at every dose, so it's not the same thing, but maybe there's something in the KIF1A population that predisposes patients to fevers. And so we kind of have to take that into account. But we've performed detailed analysis to really try to understand the cause of the fevers and to make sure that we wanted to continue treating this patient, that it was safe to continue treating this patient. So it was really helpful for us to, again, be able to base our analysis on previous knowledge that Ionis had uncovered and published and everything. So we wanted to make sure, are the fever caused due to inflammation or maybe immune activation? And so how can we try to understand this? So we wanted to assess cytokines and complement proteins in the CSF. We knew from previous work by Ionis that in monkeys, the alternative complement pathway can be activated by ASOs of this chemical class when given subcutaneously. Now, we had never seen that when it was given intrathecally to monkeys, but I mean, never say never, so we need to try and understand it. So we did the complement analysis and the cytokine analysis in our patients. Cytokines didn't show anything in the CSF, so there was no active inflammation going on. We did see some increases in the classical complement pathway, different from what we saw in monkeys, because in monkeys it was the alternative pathways. There's three different pathways for immune activation, so it was not the same one that seemed to show anything. However, it's also important to realize that, again, we kind of are at the forefront of things here. There are no published normal values for complement proteins in the CSF in pediatric patients. There are values that have been published in adults with Alzheimer's disease or multiple sclerosis or schizophrenia, but not in patients. So we do see a slight increase in this one analyte, but we don't know what the bottom and the top is of that margin of analysis, right? But again, we're tracking this and we're taking this seriously. So we believe that at the end of the day, though, this may be a novel ASO-KIF1A disease interaction that we really need to track in this patient. So from all the analysis that we've done, we have concluded that it is still safe to continue treating this patient. And what also helped is the second patient that is getting this KIF1A ASO is not exhibiting these post-dose fevers. So we're imagining at this point this is not an ASO thing in all patients. It just might be in our first patient, just reacts differently. So we feel it's safe to still continue treating the first patient, the second patient, and to initiate more treatments in additional patients. And we have done what we needed to do. We developed the monitoring plan. We shared that. We developed the monitoring plan with the treating physician. We shared that with the FDA. We gave them all the data. We said, this is what we think is going on. They agreed with us, and they said, it is fine for you to keep on dosing this patient. We just want to make sure that we're going to observe opening pressure to make sure that there's no, high pressure hydrocephalus happening. And we don't dose if the opening pressure is too high. We'll wait a little bit and then try again. We'll continue the assessment of the analytes, and we'll pre-treat the patient with steroids to help her not have those fevers. And then MRIs have been done and will be done if deemed clinically necessary. So we have also written a safety bulletin after we told the FDA. We wrote a safety bulletin that we shared with all our physicians. You can ask them. They got the safety bulletin. And what we've also done is we've put a safety update on our website. So if any one of you is interested in really reading and understanding a little bit more of what I just said today, please go to our website under safety updates. So I hope that I've shown you that, first of all, safety is extremely important to us. But safety is also not an accident. Safety in what we do is really a product of us, first of all, trying to select the most optimal ASO through the rigorous process that we go through in ASO design and discovery. We have deep experience in antisense technology. We have deep experience in clinical trials and drug development in general. We have all the committees that look at the data as well, and that really help us make sure that our thinking is, solid. And then we have the professional management of all our investigators. I can't speak enough about all our investigators. They are really top-notch physicians who really care about every patient that they treat, and I just love the collaboration that we have with them. So We have treated by far the largest number of nano-rare patients with an individualized ASO as an institution. And we have excellent safety across all the organs and using all the different routes of administration. We're collaborating closely with our treating physicians and monitoring safety on an ongoing basis. I can't repeat this enough. This is what we do every day. And we are deeply committed in sharing this information. And so with that, I wanted to thank you. Again, thank you to all our investigators who are working with us to collect all this data. Thank you to all our committee members who help us make sure we have the best ASO for your patients. And then most and foremost, again, thank you to the patients and the families for trusting us. So with that, I will invite Sarah back on stage for her talk. Thank you so much.